We are live. All right.
Should we get started?
I think we have 3 minutes, Jim.
My watch is fast. Okay.
All right. It's 6 o'clock, and I want to get everything started. So I want to thank everybody for coming on board today. It's our first virtual 2020 Nephronet Nephrology Update webinar. So we're sorry that we couldn't be live in Atlanta, obviously for COVID-19, but so we're going to host a series of virtual webinars. So you will get CME credit for this. So we'll give you some instructions on how to get that after the presentation's over. But first I want to thank, most importantly, Alexion for sponsoring this webinar. We really appreciate your support. And then everybody that helped get this up and running, Terry Smith, Trent Jones, Maggie Hunter, and Tony Gillum. Also, I wanted to tell you guys, in the bottom of your chat, there's a Q&A and a chat box. So today, if you have any questions for Dr. Tumlin and Dr. Roven, you're going to get in the Q&A box and you're going to open it up and type in your question. We'll use those at the end of the webinar to make sure that we have some interactive back and forth. So it'll be fun. So first, let me introduce our speakers, and I'm going to hand it over to them. They'll be speaking today on lupus nephritis, the role of B-cell and complement altering therapies. It's a debate on management. And first we have Dr. James Tumlin. He is a professor of medicine at Emory School of Medicine. He is a nephrologist at Georgia Nephrology and the president of Nephronet. And secondly, we have Dr. Brad Roven. He's division chair of nephrology at The Ohio State University. And so Dr. Tumlin, I'm going to hand it over to you to go ahead and get things going.
Well, thank you, Jeremy, and thank you everybody for taking time out of your busy day for this. So Nephronet's really excited about this opportunity. It's one of the upsides of the COVID virus. And I had the opportunity to work with my dear friend and distinguished colleague, Dr. Brad Roven, who many regard as one of the leading, if not the leading expert in lupus nephritis. So Brad, why don't you take it away?
Thanks, Jim. So we're going to debate, and Jim and I love to do this. We've done it for many years now at various forums. So nobody was really hurt in this, so don't worry about it. if there's any bloodshed. But as Jim said, my interest is in lupus nephritis and autoimmune diseases of the kidney. Our laboratory studies inflammation, and clinically we do a lot of trials, of course with NephroNet, to study lupus nephritis and novel therapies. So I think we'll have a lot of fun here, and hopefully everyone will learn something. So I just want you to know that throughout the presentation, this is representing me. This is Brutus Buckeye, and I have no idea what this is, but that's representing Jim. And Jim, the person who controls the information controls all. May the best mascot win.
Yes,
okay.
before you go on, Dr. Roman, I just want to remind the audience that the Brutus is a toxic nut, and let the metaphor for Dr. Roven stand alone.
And I will just remind the audience that Brutus is looking at this thing, and this is what he actually sees. All right, enough fun. Let's get going. These are my disclosure statements. I don't think they'll interfere. These are Jim's disclosure statements. They won't interfere. And I think all of you are aware that literally in the last year, there's been a renaissance in lupus nephritis therapeutic trials. And this is good for the entire lupus community, it's good for nephrology, and it's good for clinical trials in general, since we've been rather stagnant. So this is a small sampling of currently active clinical trials. I haven't maybe included everyone's favorite trial, but in the red are the trials we're going to discuss today, primarily because these were positive trials. And what I want you to see here is obinutuzumab is an anti-CD20 like rituximab, and we'll talk about that. And it completed a phase 2 trial. Of course, its target is the B cell. The phase 2 trial is done. It was a positive study. Phase 3 is enrolling. Belimumab is a monoclonal antibody against BLyS or BAFF. BLyS or BAFF, as you know, is a B cell growth and survival factor. This is a phase 3 study. It's completed. The paper is not only submitted, the paper has been published in the New England Journal, another positive study for lupus. Now, you might notice here some of these others that will be coming on board soon also look at B cells. And B cell lineage, plasma cells. Anifrolumab is rather unique, and that study is finishing up shortly. It's an interferon alpha receptor antagonist, and of course, it will affect multiple cell types. And the last one I'd like to discuss today is the voclosporin trial, small molecule calcineurin inhibitor. Its main cell type target is the T cell, of course. You're all familiar with calcineurin inhibitors, but it also has effects on the podocyte, which may be beneficial in stemming proteinuria early on in lupus nephritis. The phase 3 and phase 2 trials are done. The paper hasn't been published yet, but both of these were positive, so that's very exciting. So let me just sort of orient everyone to where we are right now. I did a survey of recent clinical trials in some of my own cohorts and some other folks' cohorts, but really looking at complete renal remission at 1 year with standard of care therapy. So I'm not looking at any of the experimental arms in these clinical trials, but using drugs that we commonly use, mycophenolate, urolupus cyclophosphamide, high-dose cyclophosphamide. We used to use quite a bit of oral cyclophosphamide, and TM Chan also did that in Asia and Hong Kong. You can see that although the rates are variable, and we have to be a little bit careful, frankly, because everybody uses different complete response criteria, but although the rates are variable, I really compiled down here, the 1 year complete renal response rate using mycophenolate is around 35%. The 1 year complete renal response rate using low-dose intravenous cyclophosphamide is about the same. So we're only hitting about a third of the patients after a year of intense immunotherapy, and I think we can do better. So let's talk about the B cell. We'll move into the meat of this discussion, the B cell targeted therapies. So the B cell lineage, of course, has B cells that go on to make plasmablasts. Plasmablasts are precursors to plasma cells, and the plasmablast does actually also produce antibodies and can produce autoantibodies. And of course, the plasma cell is the autoantibody factory. And what we'll talk about today, it will be CD20. So this is, I'm showing rituximab here, but you can just substitute in obinutuzumab. And you can see that rituximab affects the B cell and a little bit the plasmablast, but doesn't have any effect on the plasma cell, which really is the main cell in terms of autoantibody production. And then belimumab is a growth factor, actually works on the BAFF receptor, on the B cells, and again, not directly on the plasma cell. So I want to start out with a randomized controlled trial. This is the phase 2 trial of obinutuzumab paired with mycophenolate and corticosteroids in proliferative lupus nephritis. And the nickname of this trial was called Nobility. So this is the trial schematic, and there's several unique features here. First, all the patients received a target dose of mycophenolate between 2 and 2.5 grams a day, 1 gram of intravenous methylprednisolone, and a prednisone taper. Notice this taper was starting with a lower dose of steroids than we normally do, and by 12 weeks, we were down to 7.5 milligrams per day. So one thing I hope you'll take away from this is that the newer trials are using less corticosteroid, which is gonna really be of benefit to our patients with lupus. The other thing that I wanna point out, this is a 2-year study, okay? So it was 104 weeks with the double blind going. And that's rather unique too. And we did learn some lessons from earlier trials in which after 6 months or a year, we really weren't seeing remission rates. Okay, obinutuzumab or placebo was at 0 and 2 weeks, 1 gram each, just like we do with rituximab, and then repeated again at 6 months and 2 weeks after that. This trial only included proliferative lupus nephritis. Now, you could have class 5 with your proliferative, but no pure class 5. The primary endpoint, and this is a little complicated, so I'll spend a second here. Complete renal response at week 52, and then of course overall response and changes in the levels of serology. This phase 2 trial was to test to see whether there was enough data to go forward with a phase 3. And so we set a pre-specified significance level that was not 0.05 like you're used to, but 0.2. That comes into play a little bit, but really when you look at the trial as a whole, not so much. This is the population in the obinutuzumab arm, the placebo arm. They were well matched. I want to point out that we had a lot of patients of Hispanic ethnicity. Generally, these patients have very severe lupus nephritis, as you know, and so this was not a trial of mild disease. And we really do want to encourage diverse participation in these trials. We had about 10% Black patients. We need to do better on this in our lupus nephritis trials for sure. The mean proteinuria was about 3 grams in both groups. About half the patients had double-strand DNA positivity, and about 70% of the patients had a low C3. So, just to get right to the meat of the argument, this is the complete renal response rate and the overall renal response rate, which is complete plus partial. And this is week 52. This is week 76. And if you come in on the ASN in a couple weeks, we will present the actual 2-year data, which I'm unfortunately not allowed to share because it's embargoed. But anyway, suffice it to say it works. I'll just leave it at that and without violating anyone's trust. So at week 52, we had a greater number of patients, 35% versus 23%, being treated with obinutuzumab plus background therapy who achieved complete renal response. That delta of 12% yielded a p-value of 0.12, which was below the 0.2 that was specified. So this was considered significant and a go signal to move to phase 3. So that's good for all of us. But if you look now at week 76, which is a secondary but critical secondary endpoint, p-value 0.01, the response is up to 40% in the obinutuzumab group as compared to the placebo group. And when you consider overall response, they were all significant at both time points. This is the urine, this is the complete response criteria. The urine protein creatinine ratio below 0.5. You had to be within 15% of the patient's baseline serum creatinine, and you had to have no reds or fewer than 10 red cells and no casts, of course. What I want to point out is look at the actual time curve of complete renal response. And these are the two, of course, infusions of obinutuzumab. But you can see here that the patients really start to separate at 6 months. Again, I want to emphasize this, and I know Jim will emphasize it. It takes a while for most B-cell therapies to kick in. So that's another point to take home. But after that, it sort of looks like the complete responses in the placebo group after a year and a half start to drop. So I assume patients are starting to relapse, and this has a continued upward trend. Again, a good sign. And this is the safety summary at a year and a half. And what's really critical is that there were no increased safety signals or adverse events in the patients given belimumab on top of MMF or MMF plus corticosteroids. And importantly, if you look at the deaths, there was 1 death in the obinutuzumab group. It had nothing to do with the drug. There were 4 deaths in the placebo group, and none of these, except perhaps this last one, respiratory infection, had to do with immunosuppression. PML probably had to do with prior immunosuppression. Serious adverse events were similar. Serious infections were actually higher in the placebo group. All right, Jim, take it away.
Okay, well, Brad, that was a terrific presentation. I just want to congratulate you on being granted parole last month. I'm sure your family's glad to be with you. Next slide, please. So what we want to talk about is how good is the obinutuzumab data compared to rituximab? So let's go back over the original. This is Ellen Gisler's paper along with you on the original LUNAR trial. So very similar study looking at MMF plus placebo with steroids, MMF plus rituximab. And the complete renal response, as noted on the left, was about 30.6% in the MMF plus placebo group, and only 26.1 had complete response with rituximab. Partial response was a little bit better. So in MMF, it was about 15% going up to just under 31%. And as you pointed out earlier in your talk, there was a high degree of non-response, which is sort of what we really need to be talking about. Next slide. Now, if you put this next to the obinutuzumab data, well, it's not so impressive. So again, on the left, there is MMF plus placebo at about 31%, MMF plus rituximab 26%, but when you add obinutuzumab data, you only jump up to a value of about 35%. That number is most likely not statistically significant. If you then take the partial response, the less rigorous one, you see, in fact, that rituximab actually outdid obinutuzumab at 30.6% versus 21%. So again, this is sort of a new agent that maybe is not doing as well as the one we've had before. Next slide. And again, this is to point out that, you know, had you designed the trial better, Dr. Rovin, we would have looked at 52 and even 76 data, which you've already pointed out. And it really wasn't until much later in the course of the study that you started to see a difference between the two groups, between complete response rate and partial response rates. So to your point, and I totally agree with you, rituximab and any type of B-cell eliminating agent is going to be not only for induction, but it's also, in my opinion, going to be for maintenance and long-term suppression of these patients. Next slide. Okay. Well, I'm not done. So there's all lupus is not the same, Dr. Rovin. So let's talk about crescenteric and proliferative forms of it. Next slide. So this is from the Euro Lupus study, where they looked at the effect of crescentic lupus nephritis, which has been well established by Gary Hill and a number of other authors to be an independent predictor of progression at 5 years. So in the Euro Lupus trial, 18 patients with refractory disease had presence of crescents. And of the cyclophosphamide, azathioprine, and MMF treatment failures, 100% were of crescenteric forms of it. Proliferative lupus nephritis was seen in about 16 of 18 of these patients. Crescentic lupus nephritis was in 13 of 18, or 16%. And in the rituximab rescue patients, a full 72% were crescentic disease. Of the rituximab failures, half of them were crescenteric disease. So what this says is that when we're designing these trials, number one, we need to either include the crescents or exclude the crescents, or do a separate randomization for crescenteric disease, because it's a different beast and may require more aggressive therapy. Next slide.
So Jim, Dr. Tumlin, as usual, you're misinformed. Obinutuzumab has an advantage over rituximab. So let's look at that. This is a schematic of obinutuzumab, and Obi is a humanized type 2 anti-CD20 antibody, and it's approved for malignancies. Compared to rituximab, it's actually more potent. This type 2 binding conformation gives greater direct cell death, and it also does not allow internalization of the molecule. So one of the problems with rituximab is it's taken up by the B cells, and then it's no longer effective. And then something that's really important for patients with lupus is that rituximab relies heavily on complement-dependent cytotoxicity. And of course, complement is consumed in lupus, and you may not have enough available complement. And this leads to really sort of a mediocre B-cell depletion compared to obinutuzumab, and I'll show you that in a minute. This is a study that was done just looking at B-cell depletion in rheumatoid arthritis in patients with lupus. This is patients treated with rituximab and obinutuzumab, and you can see that there was an advantage to obinutuzumab in these patients. Now, what I want to really get to is how good is peripheral B-cell depletion. And just again, for the audience, we're using peripheral B-cell depletion as sort of a surrogate for what we think is also happening in the kidney. And we have not directly tested that, so you have to keep that in mind. But at least looking at the comparison of obinutuzumab plus MMF versus rituximab, and these are not a direct comparison. This trial was done several years before this trial, but you can see that by week 2, almost all of the peripheral B cells were depleted using a count of less than 5 cells per microliter, and this was sustained throughout the entire 52-week period. Now look at rituximab. It takes a while to get up and running. It eventually, by 3 months, sort of gets to the level of obinutuzumab. But then, even though it had the same sort of double dosing regimen, it doesn't last throughout the entire 52 weeks. Why is that important? Well, I'd like to reconsider the LUNAR trial from the viewpoint of B-cell depletion rather than the data Jim showed you, which is the exact data. That was the results of the trial. But this was a post-hoc analysis we did a couple years ago. And we looked at complete renal response at 52 weeks and at 78 weeks in patients who had complete peripheral B-cell depletion with rituximab or incomplete B-cell depletion. And you can see here that if you do that, by 1 year, you are starting to see an impressive difference between responders and non-responders, or people who achieve a complete renal response at 1 year. But this is really, here's the point. Look at the difference at a year and a half. So we still have an increase in people having complete renal responses, and here, not so much. And so I hope you caught what Dr. Tumlin said, because even though we're debating and we're on opposite sides here, I agree with him. We need to start thinking a little bit differently about induction and maintenance.
Yep.
Maybe that's not the best way to think about how to treat lupus. And certainly these data and the data that I showed you for the obinutuzumab trial really suggest this is a long-term game with a B-cell depleter.
Okay, well, before I go into my next part, Brett, I really agree with you on that statement. I think that I'm just going to give a little plug here that, I mean, I think that people that have very aggressive multi-organ involvement of lupus involving the lungs or the CNS, you know, don't count out Cytoxan. It will save a patient's life. And the long-term suppression with drugs like B-cell suppression is where I think the trend and the modification of practice patterns will go. So let me bring up a point that the normal physiology, the body's response to depletion of a B cell is to upregulate BAFF, or B cell activating factor. And this factor has its own receptor on B cell. It has proliferative effects. It causes class switch and B cell maturation, allows for interaction with T cells. So it clearly propagates an immune response. So this data here shows you the serum BAFF levels after patients have either been treated with rituximab or our old friend cyclophosphamide. And as you can see, over the course of time 1 to time 2, and I believe this was a 6-week period of time, there was a marked upregulation of BAFF, indicating that the body is responding to the depleted B cells from either rituximab or cyclophosphamide. Next slide. Now, this is a fascinating study that also tells you that not only when you're upregulating BAFF and then upregulating, trying to regenerate your B cells, autoantibodies are selectively upregulated by this BAFF pathway. So this is a fascinating study that looks at a murine model of lupus, and you can see in the upper panel of the histology, this is a normal histology of the glomerulus. And then after the animals were treated with cyclophosphamide for their lupus nephritis, they then stain the glomeruli for the presence of BAFF. And as you can see, there is a marked upregulation of BAFF, not just in the peripheral blood, but in the glomerulus itself, which is fascinating when you think about it. If you then give an anti-BAFF therapy to it, you totally abrogate that upregulation of BAFF. And you see that on the right by the serum anti-double-stranded DNA antibodies. So under cyclophosphamide alone, there's a significant rise, but then if you give cyclophosphamide with anti-BAFF therapy, you abrogate the rise. So what this, before I move on, what this is saying is that, and I think Brad's made this point before, we need to think about multiple pathways. We need to take a lesson from our oncology colleagues about blocking pathways at different points to getting additive effects with the least amount of toxicity. Next slide.
So, you know, Jim, what you suggest is relevant for mice, but we really should treat people. And we actually did the study that Dr. Tumlin is talking about. This was called the Calibrate study. It was phase 2. It was open label. And I can discuss the problems with this study a little bit, but let me just show you the data because it's sort of interesting. This looked at lupus induction for lupus, proliferative lupus nephritis, and it was a bizarre regimen, I must admit. Steroids, cyclophosphamide at this dose, a gram of rituximab. This was given at week 0 and 2, and then patients were randomized to placebo or belimumab at week 4. Okay, so you have to picture this. We're inducing the patients with steroids, a dose of cyclophosphamide plus rituximab, and another dose of cyclophosphamide plus rituximab, and then either placebo for the next year or belimumab. All right, and so the idea was, can you induce with a B-cell drug and then get benefit by blocking the BAFF increase that Dr. Tumlin just showed you? And the answer is not in humans. So this is the 6-month data, the 1-year data, and I'm sorry, I should have fixed this last night. This is 96 weeks. But anyway, this is complete renal response, partial renal response. This is the people who received rituximab and cyclophosphamide and placebo, and these are the people who received belimumab as well as Ritux and cyclophosphamide. no difference in complete renal response at week 24, no difference in complete renal response at week 48, and no difference in complete renal response at week 96, although the percentages may be starting to go up, and I want to say something about that. The same thing was true in the partial responders, and the non-responders are listed here. Now, this trial was designed and it went through the immune tolerance network, so it had to be designed the way it was. But first of all, this is sort of a goofy regimen to start with, and I think none of us would necessarily have done this. Second, the real end game in this is what Jim and I have been trying to say, which is when you kill B cells, by whatever means you decide to kill them, cyclophosphamide or rituximab, BAFF goes up. BAFF then induces B cells, and it can make them make more autoantibodies, so it can make the disease worse. So instead of looking, in my opinion, at complete and partial renal response, what this trial should have been designed as would have been a maintenance trial, because the idea would be get them in remission, and then see if the BAFF helps keep them in remission, because you're not having the BAFF surge and you're not regenerating autoreactive B cells.
No, Dr. Roven, so let me just say for your audience, before you move off this slide, to remind everyone, so Brad's get these, very bold conclusions, but if you look at the withdrawal rates in both the cyclophosphamide and the belimumab groups, it was 71 and 67%. So it's really going to be hard to know, to your point, about long-term benefit, because what this really should have been is a recurrence study.
Yeah, a maintenance trial. Yes, exactly. Yeah, okay, so I agree. There were so many people, and why did they withdraw? Because they didn't respond, maybe. Okay, so speaking of BAFF, belimumab works too, and this is an illustration of BAFF staining in the kidney in class 4 lupus nephritis. But I want to bring you up to date now on this this trial, which is the BLISS-LN trial. This trial was also unique in several ways. So these were patients with active lupus nephritis, and they could have 3, 4, or 5 LN. They were randomized to either belimumab or placebo, and then they had standard therapy. Now the background, this is one of the unique aspects of this trial, the background therapy in the BLISS-LN trial was at the choice of the investigator at the site. So you could give corticosteroids plus mycophenolate, followed by mycophenolate, or you could give corticosteroids plus urolupus, low-dose cyclophosphamide, followed by azathioprine. So this is a very unique study design. Like the Nobility study, this went to 104 weeks, okay, and that was where the endpoint was. But look here, at week 24, steroids had to be less than, it had to be 10 milligrams or less, and then that had to be continued throughout the rest of the trial. Yes, if you had a non-renal flare, joints, rash, etc., you could get a brief period of corticosteroid. The primary endpoint was also unique in this trial. It's called the primary efficacy endpoint, and instead of being complete renal response the way we know it, it used a little bit of a relaxed proteinuria criteria. And so I thought it would be useful to sort of compare the primary efficacy renal response, the PERR, and the complete renal response. Now note, the key secondary endpoint at week 104 was complete renal response, and I'll show you both. But the point is that the urine protein to creatinine ratio for the primary endpoint was less than 0.7, and that's based on fairly recent data that shows if you achieve that after a year of therapy, the long-term prognosis for the kidney is really quite good in lupus nephritis, compared to the standard less than 0.5. GFR had to be greater than 60, and here the GFR for complete renal response had to be greater than 90. GFR could not fall more than 20% below the eGFR, the pre-flare value. And then for the complete responders, it couldn't fall more than 10%. And of course, rescue therapy was not allowed in either one. So the point I'm making is that we looked at a relaxed endpoint that may actually prognostically be quite good. And we also looked at complete renal response. Again, a unique aspect for this trial. And then we also looked at renal-related events like end-stage renal disease, doubling serum creatinine, and flares or renal worsening. So here are the top-line results. This is the PERR at week 104. This is the complete renal response at week 104. And you can see that this was higher and significantly higher in the belimumab group, both for the PERR and also for the complete renal response. And here are the p-values. This is sort of a graph of when these endpoints were achieved at each visit and the number of patients. And you can see here, again, this is taking close to about 6 months to start to see a separation between the two groups. And the same thing, it may be a little separation earlier in the complete renal responders, but this is again, showing you that B-cell drugs take a little bit of time to work. Here's something that I think I really want to point out, because this is rather important for us to understand. And frankly, this may be one of the more important considerations of the trial, because the difference, the delta between the placebo group and the belimumab group in terms of complete renal response is significant, but it's on the low side. But look what happens to EGFR. EGFR sort of improves in both patient groups initially, and then over the 2-year period of time, EGFR is sustained in the belimumab group, and EGFR tends to deteriorate somewhat in the placebo group. Coupled with that, these are the renal-specific data. This is looking at, again, placebo and belimumab. In terms of renal worsening, so in other words, increased urine protein to creatinine ratio, decreased GFR, and you can see for all of these, it's really more in the placebo group than the belimumab group. Again, giving the idea that this keeps the patients stable and hopefully not flaring. Now, fortunately, there were very few doubling of serum creatinines or deaths or end-stage kidney disease, so that was good. And then the addition of belimumab to either cyclophosphamide or to MMF did not increase the adverse event rate. You can see that in both groups, 11% and 10% had one severe adverse event. Infections of special interest, so opportunistic infections, etc., were pretty well balanced between the groups, and deaths were well balanced between the groups. So again, something that seems to be emerging, at least to me, is that the addition of B-cell therapies, when we want to start talking about multi-target regimens, seem to be added with not complete safety, of course, nothing is completely without an adverse event, but we can do this and we can be relatively confident that we're not harming the patients. Okay, these are some final thoughts I had on targeting B cells in lupus nephritis. We've been through this a couple times. Anti-CD20 therapies act more slowly. BAFF therapy showed superior efficacy during the course of treatment, but also maintained a superior response during long-term follow-up. This is key. And again, I can't give you the data, but we're presenting in-depth look at belimumab, the results of this on the kidney, especially for nephrologists at the ASN. And if you can tune in, I think that would be a lot of fun, because you'll see some real comments on this. And then this is what Jim and I were talking about. If we abandon induction therapy terms and maintenance therapy, we should think about treating lupus nephritis, I believe, on a continuum. We have to attenuate inflammation early, and then we have to correct the aberrant immune response. And targeted B-cell therapies may be useful when they're started early, but you expect them to target the autoimmunity rather than help you with the induction and control of lupus nephritis right up front. Okay, Jim, take it away.
Okay. All right, so now we're going to switch gears. Oh, thank you, Brad. Switch gears a little bit back up, please, to talk about a new agent that's come along, which is voclosporin. Most of you probably heard about this. Voclo is actually it's the second or maybe third generation CNI that's being developed by the Arunia Corporation. It's a very interesting molecule. And this is the data from the Aurora trial, which was just completed, and the paper is being submitted, as Brad indicated. And it was a comparison of mycophenolate at 2 grams a day with a rapid corticosteroid taper, and I'll go into that in a second, comparing placebo to 23.7 milligrams twice a day of voclosporin. That was derived from the phase 2 trial that was presented 2 years ago. Like the current trend, this is for everybody in the audience, we need to get away from all these steroids. And so in this study, they went from roughly 20 to 25 milligrams per day at week 1, all the way down to 2.5 milligrams by the end of 4 months. This was a composite endpoint, and it's really important the audience understand it, because a lot of really learned people would say, well, if you just simply look at proteinuria, we know that a CNI reduces proteinuria, but this study is not just that. So it was the definition that Brad talked about just a moment ago, 500 milligrams per day. But also your GFR had to be above 60 with no more than a greater than a 20% reduction over the course of the 24 weeks. You could not have any rescue medications, and you had to be less than 10 milligrams a day of oral prednisone. Next slide. So as you can see here, again, if you look at the control group, so this is MMF and prednisone alone, it's basically superimposable to the data from the Lunar trial, about 22.5%. And, you know, frankly, guys, Brad and I talk about this all the time. I don't know why we were always satisfied with a 22% response rate. I mean, that's just really not that good. But when you added the voclosporin to the regimen of the MMF and steroids, you went up to just under 41% response rate. And that was the primary efficacy endpoint at 52 weeks. Next slide. Now, they did a further breakdown this on race and ethnicity. So they looked at Asians, Caucasians, African Americans, and mixed race individuals. And if you can look on the Asians, which also can have very aggressive disease, there was a complete response rate, a renal response rate of from 17.9% up to 41.5%. The p-value of that was highly significant, p.005. This is a lot of what the data that T.M. Chan had shown in his group. Interestingly, in this analysis, and again, this is a post hoc analysis, the Caucasians did not reach significance on this, although it was clearly a trend, but the p-value on that was 0.165. However, in the arguably the most aggressive subpopulation of patients, people of African descent, there was a highly significant value of p.045 from 15 up to a 46% complete response rate. Mixed individuals was less notable. Then if you look at the Hispanic group versus the non-Hispanic groups, you see a similar trend. So again, if you look at renal response rate versus partial response rate, there was a sparing in the Hispanics. So the addition of voclosporin raised the rate from 18, almost a doubling, to 38%. A partial response rate went from 52% up to 68%. And in the non-Hispanic population, you saw a similar trend on renal response rate and complete response. So again, this was really quite a very successful study. And again, I think did well to do away the idea that, oh, this is just another CNI. And I'm going to explain why it's not. Next slide. So if you look at the adverse events, I'm not going to spend too much time on this, but they were very well matched. What was interestingly was the discontinuance rate was actually higher in the placebo than it was the Voclo group. I will tell you that there was no difference in electrolyte disorders as well, and I'll come back to why that's significant in a moment, but the drug was very well tolerated. Next slide.
So, you know, to create some controversy, you know, Jim, okay, so here's a brand new drug. Why would I use this new drug when tacrolimus and cyclosporin work just as well? And so let me just reintroduce everyone to the original multi-target trial from China. This was a large trial, 360 patients. Everyone had 1,500 milligrams of methylprednisolone, and then started with a steroid taper. They were randomized to NIH cyclophosphamide or MMF plus tacrolimus, fairly low dose of both, and complete renal response was the primary endpoint at 6 months. So here are the top line results, and I realize the table's a little hard to read, but if you look here, just at the red boxes, overall, this is all the classes put together. The multi-target regimen had a 46% response rate. That's pretty darn good at 6 months compared to intravenous cyclophosphamide, which was 26%. And of course, this was significantly different. Now, I do want to point out that even though the idea of the original Chinese study was to add 3 drugs, use them in lower doses, and decrease toxicity, I'm not quite sure that that was accomplished. In the multi-target group, this was not statistically significant, but you can see that serious adverse events were higher in the multi-target group. And then you can look at the various events here. And then also here, more patients in the multi-target group than in the cyclophosphamide group dropped out as a result of adverse events, about 5.5 versus around 2%. And that was not significant, but the trend was there. I guess my message from this is that even though people want very safe therapies, the addition of 3 drugs, these 3 drugs in particular, is still something that causes toxicity, and that cannot be ignored or forgotten when you're treating these patients. So I do want to point out that in long-term follow-up, the patients in the Chinese study from the original cohort who had a response went forward in time. The cyclophosphamide with azathioprine maintenance and the MMF TAC with a reducing dose of tacrolimus and mycophenolate. And of course, this is where they start out, and we know that in the multi-target group they had done better. But you see very quickly in follow-up that the cyclophosphamide group catches up. So this was not a maintained difference over a lengthy period of time. I'm going to skip this, and I'm going to let Jim get to a really important point here.
Okay, well, thanks, Brad. Yeah, so it's a legitimate question. So we've got tacrolimus, we've got cyclosporin, we have 40 years of experience. What makes Voclo different, in my opinion? Next slide. What's been fun for me personally is that this goes back to some work I did with Emory with my colleague Jennifer Gooch. And if you look on the left-hand side, there's the 2 structures of Voclo and cyclosporin. They are identical except for a single double carbon bond where there's a red circle that you see at the top. That is the location where the molecule binds to its immunophilin binding domain, cyclophilin 16 or cyclophilin 40. Next slide. And that change dramatically alters the biology of the drug. It gives you a very predictable PK and PD, such to the point where you no longer need to do waist-based dosing. You don't need to check trough levels anymore. It has an improved lipid profile. There is a significant reduction when compared to tacrolimus of the induction of type 1 and type 2 diabetes. And it doesn't interfere with mycophenolate metabolism. That's one aspect, but there's the other exciting part, next slide, is the biology of the molecule itself. As I said before, that when Voclo or cyclosporin binds to its receptor, cyclophilin 16 in the red, it forms a heterotrimeric complex with calcineurin, which blocks the dephosphorylation of NF-ATC, which, if you remember your biology, is what causes stimulation of virtually all of the major cytokines. This subtle difference in the binding domain of cyclophilin 16 alters many aspects of the drug. Next slide. One, it makes it, next slide, it makes it 4 times more potent than cyclosporin, plus it does this. If you look at the different calcineurin isoforms, alpha and beta in the catalytic subunit, if you knock out the alpha subunit in the mouse, you get significant toxicity of the kidney. It looks like, and there's some preliminary data to support this, that voclosporin may be a more selective beta antagonist. Now, what that means is that you would get the advent of immunosuppression and hopefully with less nephrotoxicity. It's still being worked out. It's not fully established. But what I will tell you, in both the phase 2 and the phase 3 trials, one of the major side effects of CNI toxicity, hyperkalemia, was not seen in the Vocalo group. It's indirect evidence, but very encouraging. Next slide. So in conclusions, TAC and MMF are superior to cyclosporin and lupus induction. Voclosporin and MMF was superior to MMF and lupus induction. Long-term survival studies need to be done, and particularly with regard to fibrosis of the kidney, which we all worry about with CNIs. CNI toxicity needs to be evaluated, and post-treatment histology needs to be evaluated, and then looking at this agent with steroids, MMF, and other agents, and possibly even B-cell agents. Next slide. So a final thought on this, Brad, I think you're going to take that.
Sure. So these were the trials I showed you at the beginning, and this is sort of where we are right now. And maybe there's a little bit of an increase in the complete renal response rate, but I want to just a little bit of word of caution. I think you need to read this literature, and then you have to look at the criteria for complete renal response. It would be really nice if we were smart as a community and every one of these trials had the same endpoint, but we didn't, and we don't. And even though Jim and I are trying to design trials so that you can compare between one and another, you really have to take into account the criteria used for complete renal response. And I'll put one more plug in. It's not simply the complete renal response that's important at 12 months or 18 months or even 2 years. What's really important is what happens to the patient several years down the road, because these are mostly young women and we don't want them to wind up with renal replacement therapy. So I think we should start looking at what happens in these long-term trials to GFR, and we're starting to get the drug, the pharma, to look at that with us. So I think that's it, Jim. And we're open for questions.
That's great. So I hope the audience will join in and type in some questions. You can actually call in. Is that correct, Jeremy? Can they do that?
No, so they'll go to the QA link, and actually I'll encourage Dr. Tillman and Dr. Rovin to open up the QA link. So we've got 3 questions thus far. Dr. Zerlip was asking earlier, so why do you feel that drugs targeting CD20 and BLIS were positive when used separately but in combination failed?
I'm going to let Brad start with that one.
Okay, so I think, again, the combination of the two looked at the wrong endpoint.
I agree.
And in fact, when we were designing the obinutuzumab trial, we designed it, and I don't want to use the word we, which means me. I did not want to design it the exact same way as the LUNAR trial because the LUNAR trial failed. My impression all along is that the B-cell therapies really are more targeting autoimmunity than they are targeting anything that needs to be treated quickly to save renal parenchyma.
Agreed. Right.
Which is inflammation. Okay. Yeah. So in other words, if you had looked at the combination of belimumab and rituximab, I wish they would have designed the trial to have a standard sort of initial treatment regimen, plus or minus belimumab, and then have gone out 2 years or even longer to see the flare rate in the one arm versus the flare rate in the other arm. And my prediction would be that the patients in the BLIS arm would have maintained their remissions and actually, again, potentially have maintained their GFR and not lost GFR over time. So I think the design and the expectation of that trial was wrong. I think the idea was right. How about that?
Good question, Harold. Jeremy, next slide. Yes.
So Nader from Lexion is asking, so is the idea that people stay on voclosporin long-term, or will they be tapered after 1 year?
Great question. I'll take that one. That as you know, the normal practice for tacrolimus or cyclosporin is to get them into remission and taper. And as you probably also know, that for either drug, the recurrence rate of the disease, FSGS, membranous, lupus, whatever, is about 80% within 2 months of it. So there is a manipulation and sparing a podocyte function that is a calcineurin dependent process. And when you block that process, you increase the viability of the podocytes. Now, the problem with the long-term therapy is that, as you know, that within 2 years, 20% of the people are already showing signs of interstitial fibrosis and tubule dropout. So if it's true, and I want to make sure that this is not established, if it's true that Voclo is a more selective beta antagonist and therefore not inhibiting the alpha isoform of calcineurin, we would expect less fibrosis, less nephrotoxicity, and the ability to leave people on this agent for months, if not even years at a time. That's the hope. Not proven yet, but there's some encouraging data.
And Jim, you had sort of hinted at this a little bit, but let's anticipate going forward in time on the basis of these large phase 2 and 3 trials that some of these drugs get FDA approval. It may be interesting to look at various combinations of B cell and then of course the T cell antagonism with the calcineurin inhibitor. These might be things we should start thinking about now and planning in anticipation of hopefully getting some of these agents approved
I
for
agree.
use.
I think it's a very real possibility that with the majority of lupus patients, some combination of these agents that are now all coming into focus may lead to some really substantial and long-term remissions in young women. Jeremy, you got another question for us?
So can you comment on biomarker changes such as anti-DS, AB titer, and complement level in obinutuzumab trial and the LUNAR trial? And then also the belimumab trial, does that correlate with response rate?
Well, in the LUNAR trial, so basically, in the LUNAR trial, the drop in double-stranded DNA was actually, and the improvement in complement actually paralleled in the rituximab and the placebo arm, which was a little bit unusual. But in the obinutuzumab trial, and I don't show it here, but I will show it at the ASN, there was a complete and distinct drop in the double-stranded DNA levels and a rise in the complement levels that was significantly better in the obinutuzumab group, and it did correlate with sort of that time course of improvement. So again, I think that might have to do with the better, I mean, we all think that, rituximab is a great B-cell depleter, which of course that's what we knew. But now you use obinutuzumab and you can see these B-cells are gone for a long time, which is sort of scary, but apparently at least on these trials, the safety wasn't worse. The point is that I think because of the better B-cell depletion, we're starting to see the serologies correlate in the way we expect with remission. So yeah, so sometimes biomarkers don't work out as I know.
Let me weigh in this, and this is actually a genuine point of disagreement with Brad. So it take you all the way back to the late 1990s, early 2000s. There was a company called La Jolla Pharmaceuticals, which is now a completely different company, had a small molecule that was able to neutralize and totally drop by more than 90% anti-double-stranded DNA antibodies. Now, people got into this whole thing about the FAR assay and high-affinity antibodies and all this stuff. The bottom line was when they dropped the anti-DNA levels, there was no difference in the recurrence rate with recurrent lupus nephritis in the group that had low anti-DNAs. So while he's right, there's a correlation. All the glitters is not gold, and correlation is not proof. And so do I think that it's important about the complements? I absolutely do. I'm not so sure what I believe about the anti-DNA.
Okay, no, let me defend myself because I don't think we actually disagree. I think we probably agree if you interpret it the way I'm interpreting it. I am not.
Of course, right.
Well, let me rephrase this for the audience. He should interpret it the way I'm interpreting it. That's really what I'm trying to say. The double-stranded DNA, I completely agree with you, is not necessarily the pathogenic antibody that has to be eliminated in lupus nephritis. I'm using double-stranded DNA as a marker, if you will, a surrogate for immune complexes in general. Those have to go away. So how about that?
I like that.
Okay.
Jeremy, I think we have time for another couple questions.
Absolutely. Yep. I want to remind everybody, we'll continue on and answer these last questions, but to get credit for this for CME, we have to send you guys an evaluation after this. So you'll receive that via email. Just shoot that back and you'll get the credit. But let's move on to the next question, what was the median time to B-cell return in the obinutuzumab trial?
Yeah, that's a good question. So I know that you remember that was 96% depletion, right? All the way through 1 year. The next measurement I have that the company gave me was the B-cell depletion at week 104. So between 52 and 104 weeks, almost everybody recovered their B cells. Your question is more specific, which is, after you stopped after a year, which was really, when you think about it, at week 102, remember the B cell drug was given at the beginning and at 6 months. You want to know maybe at week in between, did they recover, right? And I don't know the answer to that, to be honest. And that is a good question, and I can see if I can get those data. And I don't know if you're going to have a follow-up on your website for answering questions, Jim or Jeremy.
We will
but
indeed, right?
I can try and do that. Yes.
We can absolutely do that. So a couple more questions. So in regards to the CSA side effects in women, which are cosmetic, gum and hair loss issues, do you anticipate the same with voclosporin?
Good question. I really can't tell you. So obviously in an animal study, hirsutism is not something that is easily studied. And so I don't know if that is an alpha phenomenon or not. My suspicion is that it would be. Just to digress a little bit, what's really interesting to me is that the calcineurin dependent pathways that lead to podocyte apoptosis and detachment, for that matter, they are apparently beta isoform dependent. Well, that's just perfect. Which, if you have a selective drug that is a beta antagonist, you're sparing podocyte function as well as viability. And you're also manipulating, as Brad indicated, both cytokine production through NF-kappa beta pathways as well as T-cell interaction. So the hope is that will be, that would be really a perfect drug in many ways for me. So I don't know the answer to your question. Hopefully it's less for women, yeah.
So we've got one more question and then we'll conclude our webinar. I want to thank everybody for joining. We had quite a big response and we really appreciate everybody. We've had over 100
we sure do.
up and maybe even more for the West Coast. So the last one's from Dr. Phillip as well, and says, in the belimumab trial, the patients who got MMF appeared to have responded, but those who got, appears to have responded, but not those that got Cytoxan. This suggests that possibly worse disease did not respond, ?
Yeah, that's a really interesting question. And we did, that was actually brought up in a editorial as to whether or not the patients who entered the belimumab trial where the investigator chose to use cyclophosphamide were worse or sicker. And we don't exactly know the answer to that question. I'll just tell you my experience, which was I'm a cyclophosphamide person as opposed to MMF. And so I entered all my patients in the Bliss-LN trial, and they were all on Lupus cyclophosphamide. It didn't matter that they were sick or not. So I don't know that that's true. It's a valid concern, but your point is well taken. When you break up the subsets,
Yep.
into the calcium or into MMF versus cyclophosphamide, the patients where we actually saw the difference were the MMF treated patients, not the cyclophosphamide treated patients. And I will emphasize that in the ASN. So, if you say, Brad, how would you use this drug? Well, I got to tell you, I would combine it with MMF, and I probably would not combine it with cyclophosphamide. So I'll leave everyone with a last thought from me anyway, is now that we have these new drugs coming out, we have to read these papers carefully and try and decide how we're going to use these new drugs. Because not everyone's going to respond to Voclo, and not everyone's going to respond to Obi, and not everyone's going to respond to belimumab. So I think we have to now start thinking about, where do we place these drugs in the therapeutic armamentarium? And that is actually quite difficult. And yeah, I've been giving it a lot of thought lately. I'll give some thoughts at the ASN on that.
Jeremy?
That concludes. Anything else, Dr. Tillman, before we leave?
Well, just want to put in I just want to thank everybody for your support in Nephrinet. And just tell everybody, this debate's going to be archived on our website, so you can go back over many times you want. As Brad asked, there will be some questions that will be addressed further. If you have some more on there, Brad and I will be checking in regularly with that to answer those questions. Thank you again so much, and we hope to see you in 2021 in Atlanta, Georgia for Nephrinet 3 meeting. Thank you very much.
Face-to-face.
Face-to-face.
All right. Thank you, everybody.
You'll get the evaluation sent to your email, and we look forward to seeing you. If you missed anything, you can join us at 9 p.m. Eastern. Thanks.
All right.